During interphase, a stage before meiosis, you start off with a pair of homologous chromosomes in the diploid parent cell, and they then duplicate. So you have a pair of sister chromatids, and both these chromosomes are homologous to each other, meaning they're similar in size and position. The cell has gone through interphase, meaning G1, S, and G2 phase, it is ready for meiosis.
Okay, now we've entered prophase 1. This is where the chromosomes begin to line up in a process called synapsis. Since they're lined up homologously, they'll begin to exchange genetic material with each other, causing sexual variation in offspring, and then also, the nuclear membrane begins to degrade, and spindle fibers will start to perform, preparing for metaphase 1.
So in metaphase, the two chromosomes that are in a synapsis, they attach in the middle and spindle fibers attach around them. And so following that, in anaphase, they begin to split and go towards the end of each cell.
All right, during telophase is when the spindle fibers have pulled the homologous chromosomes to each side of the cell so far that now two different cells are starting to form. These cells are now going to be two haploid cells, which each still consist of sister chromatids, but the haploid means that it's now just N instead of 2N. Now, during cytokinesis is when the membrane will completely split off, and you'll have two haploid sister cells that are identical, but they're separate.
In prophase 2, the nuclear membrane starts to disappear, and the spindle fibers begin to form. All the other steps are very similar to prophase 1, although crossing over and synapsis do not occur.
Similar to metaphase 1, the chromosomes line up in the middle at the metaphase plate. And now the chromatids are not identical since they went through crossing over in prophase 1. In anaphase, the two chromosomes begin to split from the two sister chromatids begin to split from each other and go to opposite sides of the cell.
Okay, much like in phase 1, telophase is the chromosome separating into two identical sister cells. All these cells are completely haploid but still identical to each other. In cytokinesis, these cells will completely sever, making 4 independent cells.
So now the finished product is 4 daughter cells that are each haploid, and these are made so that gametes can reproduce, and when they come together, it would make a diploid cell again. Each of these are genetically different because they've taken info from both the maternal and paternal chromosomes. This is just all for reproduction purposes.